So before I start,
So the one thing that I forgot earlier is the slide showing you my hometown and what it is in Spain, so you're going to visit, so now you know where you're going.
See the arrow? Right there. See that? Yeah, the arrow, yeah, that's the thing called the... So, our course is coming to an end. It's been incredibly exciting. We have one more topic that's going to kind of merge a couple of things. So when you go out and look at molecular data, as Wolfgang has shown you, you can basically do a genomic scan independent of any trait and basically ask, are there molecular signatures for either recent or repeated historic selection? I'm going to address the complementary thing. You go out and you look at a population, and what you see is that population has diverged over time. There's many ways we can look at this, I'll talk about in a second. So the question is, how can you determine whether that divergence, or I should say, what do you need to determine whether that divergence is due to drift or something else? As the topic here is test for neutrality in trait evolution. So in contrast to the molecular data, now what we're doing is we're looking at a specific trait. The advantage of the molecular data was it's trait independent. The advantage of this is you don't need, we'll show various levels of tests, but you don't necessarily need molecular data to do these sort of tests. These notes are on the web, by the way. And more importantly, there's a longer chapter, chapter 12, also posted as a PDF that goes into detail. I'm going to be skimming through a bunch of these, goes into long detail, lots of worked examples, and lots of the original references. Here's the overline. I want to talk about trait divergence under mutation and drift. I want to talk about two tests that Russ Lande proposed. He has a drift-only test, which is often called the constant variance. Constant variance means the additive variance isn't changing from mutation. This is good over short time scales. For example, t roughly less than 1 over the effective population size. Over long time scales where mutational input's important, he has his mutation drift equilibrium, or MDE test. Then we'll talk about Brownian and Ornstein-Uhlenbeck models for drift, Brownian and drift under constraint, Ornstein-Uhlenbeck. We'll talk about comparisons of FST and QST. This is a popular class of tests. Well then, so if you go down here, these tests are only based upon phenotype and some estimate of the heritability or the additive variance. These tests require marker data to estimate FST and genetic data to estimate QST, so this requires more information. If I have QTLs from a cross, for example, we could use methods to basically look if the pattern of QTL effects is non-random. And finally, we'll look at divergence of gene expression. Gene expression is kind of the new trait that a lot of people are working on, because in a single individual I can score 20 or 30,000 traits. And simply looking at the level of gene expression is a quantitative trait. I can measure it. We can map QTLs for that. So we'll talk about how some of these tests are applied to this. So as I mentioned, if you look at traits, they can diverge, right? And so tests for whether an observed amount of divergence in a trait mean is consistent with drift appear in a number of very different settings. Number one, I could look at divergence over a single population. So one of the first applications of this was Russ Lande took a fossil time series and asked, no genetics, can we from that fossil time series ask if the amount of change was more or less than we expect from simply drift alone? You can also look, this is ecological settings, divergence between a set of contemporaneous populations. You can look at the pattern of divergence of fixed QTL alleles, and like I said, you can apply these tests to gene expression. So this category of tests, which includes several different ones, often unrelated, has been applied from paleontology all the way up to genomic analysis. So it's a very interesting set of tests and covers a very wide range of subjects. The bottom line is whenever you see a pattern, drift should be tested and then rejected before you offer selective explanation. So the default is, if you just simply have a trait, its mean is going to drift over time because the underlying alleles that underlie it, if they're neutral, drift over time. And so you have to basically rule out the neutral evolution model before you can then put your favorite selective explanation for it, which of course will be wrong. So let's now look at the underlying ideas here. Genetic drift changes the frequency of alleles, and in particular, it changes the frequency of alleles underlying traits unless there's some other force to counter it. So because you change these allele frequencies, that in turn means the mean changes. And a very simple model for that is, let's imagine I have an additive locus. So I have effect 0, A, 2A for my 3 genotypes, where the frequency of the each copy of big A gives you an amount, an increase in the mean, and we'll let P be the frequency of that. So with this simple model, you can show that the mean is simply twice A times the frequency. The variance in this, well, if I just simply compute the variance, that's the expected value of that squared minus the square of the expected value. That's just the definition of variance. I can pull out the 2A. And what I see here is this quantity down here is the drift variance in allele frequency, right? Simply, what's the expected, starting with some initial value, what's the expected variance in P of t? And what you can show is that equals the initial frequency. By the way, I should notice that PowerPoint is really bad for dealing with double subscripted and superscripted things. So you'll notice down here that I use V of P, but over here I have sigma of P. If I do this in PowerPoint, those aren't stacked and looks awkward. So whenever you see a sigma in the tech font, that's the same as a V. This would be VP, this would be VB. So basically, this is the drift variance. So you see it depends upon time, the effective population size. That should be NE if you want to be formal about it. And the initial frequency, you just plug that into there, and therefore you get the variance between means. That is, if I take a series of observations and I look at the means, how much variance do I expect to see between those mean values? Here it is, where I have some initial founding population for each of them, and we often ignore this. Then I get this term here, and I can express that basically as twice the inbreeding coefficient. This is the inbreeding coefficient. So this expression here simply says, if I have a big population, I sample and make a series of subpopulations, and then let those simply run under drift. What happens is I take that variation that's there and randomly partition it from drift. This ignores new mutation coming in. We'll talk about that a little bit later on. So this is kind of an analysis for relatively short time scales. It is on the order basically of less than n. You'll notice, by the way, that as t goes to infinity, f goes to 1, and this thing basically then goes to the between mean variance stabilizes at twice the initial additive variance. And again, we don't usually worry about this time scale because of this, when we get this large mutation comes in. The bottom line is this. We have an expression that can basically predict what the between group variance is going to be, and it's simply a function of the amount of inbreeding, and that's the effective population size times time, times the initial additive variation. So if I just simply know the initial additive variation, T and NE, I can get an expected divergence in terms of the variance. Then I can basically ask if that's either too large or too small, given my estimate of NE and T relative to the pattern I actually see. Okay. People on board so far? Any questions? This is a technical aside, but it's very important if you're going to analyze a selection experiment. Suppose I have a time series and I'm following a single population over a series of time points. What drift does, it creates correlations among those time points. So for example, the mean at time t prime, at t, and the mean at time t prime in that same population, where t is less than t prime, is this. So the net result on here is, number one, the variance in the mean changes over time. This changes over time. Number two, different time points are correlated. If I simply did a regression of mean on time, I now have heteroskedastic residuals. Why? The variance gets bigger with t. I also have correlated residuals because of this. So because of that, if you're trying to analyze how a population changes over time, you've got to use general least squares, not ordinary least squares. And this has implications when you do selection experiment analysis. I'm just calling that to your attention. The chapter 18 online talks about the implications of this. So just be aware of that. Common mistake is people just fit, if they got a population trace over time, is they just simply fit a normal regression through it, ordinary least squares. And that gives you the right slope, but it gives you the wrong error variance. It makes the error variance much tighter than it actually is and allows you to overinterpret those results. So I talked about divergence. Let's talk about some measures of how a mean can change what we'd use. So for two populations, I can simply use the absolute difference between the two means. So mean of population 1 minus mean of population 2, take the absolute value. That's D. For L populations, I can take the between group variance. This is the expression we got before here. These are the same things. And that's just simply the mean value for population I minus the overall mean squared divided by 1 over L minus 1. This is just simply a sample variance where now my individual units in the sample variance are the means itself. So if I have 5 populations, I'd have 5 values for the 5 populations. I'd subtract each from the overall average mean and compute a variance that way. You could use this approach. A slightly better approach is to use analysis of variance. Why? Analysis of variance takes into account that when you estimate the mean for population I, you estimate it with error. And that error depends upon the sample size and sub naught. So you can basically just express that in the same way. This is the mean squares between minus the mean squares within divided by the sample size. So typically, people use ANOVA to estimate VB, although you can estimate it here. The theory still holds, this is just a slightly better estimate. Finally, let's just connect an absolute difference in means with the variance. You might imagine there's a connection, and there actually is. It's pretty straightforward. If I have, let's look at the between group variance when L equals 2. Well, L equals 2, that's just 1. This is y1 minus y bar plus y squared plus y2 minus y bar squared. Y bar is y1 plus y2 over 2. So first value, second value, a little bit of algebra shows that's d squared over 2. So you get the nice connection then that twice the between group variance is d squared over 2. And if d squared, if I give you d squared, the between group variance is simply d squared divided by 2. So those are often used interchangeably. They go back and forth in using this. Okay. So measures of divergence, either an absolute difference or a variance. Final thing, so how many of you've probably heard about chi-squares and F distributions and stuff, but let's actually talk where they come from, because it'll be a little bit important. If I have a random variable that follows a normal distribution with mean 0 and variance 1, that's called a unit normal. If I square it, so let's say the value is minus 2, I square it, I get a 4. The value is 0.5, I square it, you get 0.25. The distribution of a squared unit normal is a chi-square with 1 degree of freedom. If I sum up n of those, I got a chi-square with n degrees of freedom. That's where those degrees of freedom come from. The expected value of a chi-square with n degrees of freedom is just n. Thus, the expected value of a chi-square divided by n, its degrees of freedom is 1. The ratio of 2 scaled chi-squares is an F distribution, right? So we talk about a numerator and denominator degrees of freedom. A special case of this is if I take d to infinity, this becomes a point mass. Basically, this thing goes to 1 as d goes to infinity. Why? Because you're centered on the point mass. So a scaled chi-square is an F with n numerator and infinite denominator degrees of freedom. Question? So let's do a couple things. Explain what again? Because the whole slide or what part? Here. So degrees, it's simply this. What's the difference between a chi-square with 2 degrees of freedom and a chi-square with 3 degrees of freedom? For a chi-square of 2 degrees of freedom, I take a unit normal, I square it. I take another unit normal, I square it. I get the distribution of that. That's 2 degrees of freedom. For 3 degrees of freedom, I take 1, square it, 2, square it, 3, square it. I take the sum of 3. So if I take the sum of n of these squared unit normals, I get a chi-square with n degrees of freedom. That's just the definition of it. That's why normality and chi-squares are closely connected. The expected value of a chi-square divided by its degrees of freedom is 1. The ratio of 2 scaled chi-squares, that is chi-squared by the degree of freedom, is an F. This thing has expected value 1. Why? That's got expected value 1, that's got expected value 1. We talk about numerator and denominator degrees of freedom. And a very special case is if I simply want to write a chi-square divided by its degrees of freedom, I can write this as Fn, and now this becomes infinite degrees of freedom. That just only means that's a constant of 1. Okay. Those are some of the connections that come up. So now, we're ready to start looking at some of the tests. So assuming that t is much less than n and mutation can be ignored, then I can easily construct some tests. Let's work at those. So here's the fact I'm going to use So remember here I said if I had a chi-square, a normal, unit normal, if I square them and sum them, I get a chi-square. Now let's do something a little bit different. I look at y1 minus the sample mean. If I take 1 over n minus 1, this thing here, if the data are normal with mean 0 and variance sigma squared, this thing here, n minus 1 times this sample variance, is a constant sigma squared times a chi-squared with n minus 1 degrees of freedom. So if you look at sample variances, they follow chi-square distributions. And you can use that to construct confidence intervals and do hypothesis tests. All that stuff is in that chapter 12 that you've got a copy of online. So we're going to use this. Why? Because what is that? This basically says then that the between group variance, right, which I estimate here, is basically the expected variance. So the observed between group variance follows the expected variance times a chi-square random variable. In particular, Russ Lande proposed 2 tests. This is the first one dealing with the effects where I've got populations and the populations diverge, but I'm assuming that divergence is all due to partitioning the initial variation. It's not due to new mutation. This is a relatively short time scale test. And what you can show then is the expected, just going back up to here, assume that founding size is really large, we can ignore that term. You look at this term here, you get the between group equals this expression times additive variation, and it turns out you can write that basically as this. So the between group variation at time t equals the initial additive variation times time divided by the effective population size. So his constant variance test, because he's assuming that you're not putting any new input in here from mutation, is just the observed variation you see divided by this. So if you know the initial additive variation and you have an estimate of t divided by NE, you can put this down in here. And as you might imagine, right, the ratio of these things, it provides a test statistic. So let's look at what this first thing is. There's the sample variance, right? It's just the ith mean minus the overall mean, 1 over L. Well, from this slide down here, that follows the chi-square distribution with this expression here for the between group variance. I then plug this down in there, and I get this result here. So what it says is that's the expected between group variance. It follows this times a chi-square random variable. And then what you can then show is the ratio, this constant variance test then, is just a chi-square with L minus 1 degrees of freedom divided by its degrees of freedom. That's just an F test with L minus 1 numerator, infinite denominator degrees of freedom. So don't worry about the details. Those are things you look up, right? The idea is understanding the logic. And the logic here is that we have an expression based upon time divided by NE and the initial variance that tells us how we expect an additive character to diverge over time. So we have an expectation. I can then take an observed pattern, and I can test whether that observed pattern is significantly too large, that is, the variance is significantly too large, directional selection, or is significantly too small, stabilizing selection, relative to that initial, the expected value. The expected value depends upon something that's easy to get at, the additive variation. It also depends upon time and effective population size. More generally, and we'll get to that later. So here's an actual example from Lande's 1977 paper. So Wyatt Anderson had 5 populations of Drosophila pseudo-obscura that he basically reared in different temperatures over a 12-year period, and then he raised them in 2 common environments, and then basically among these 5 replicate populations, he measured all in environment of 16 degrees, all in environment of 25 degrees, and measured wing length. So the initial additive variance in these 2 environments were 0.88. So the additive variance in these environments were 0.88 and 0.77. The between population variance between these 5, the means of these 5 lines for wing length, was 6.2 at 16 degrees and 4.4 at 25 degrees. From the year here, it's approximately 150 generations. Population size was always probably larger than 1,000. So if you take 1,000 and it's significantly different from drift, if your population size is even larger, it's going to be even more significant from drift, right? The bottom line is, if you have a small population size, drift is more important. So if your actual population size is above that, and 1,000 won't account for it, then you can reject drift. So you basically form the constant variance test. There's the between group variance, there they are, 6.2, and then 4.3. Down here is time 150. This is the initial additive variance, 88 and 77. That's the effective population size, and you get whopping big numbers, right? This thing here is compare this number, this is for the 5 populations at 16 degrees. That's a chi-square with 4 degrees of freedom numerator, infinite denominator. The chance that it's bigger than 4.6 is 0.1%. That's a heck of a lot bigger. So both these are very highly significant. So therefore, there's good evidence to suggest this divergence we see in temperature over the 150 generations. You'll notice 150 over 1,000 is still pretty small. We're still in the thing where we're partitioning initial variation. So there's strong evidence that this divergence we see in laboratory populations was not driven by drift, but rather was driven by something else. There's too much between group variances. Remember, the 5 populations raised consist of 2 that were raised at 25 degrees for 12 years, 2 at 27, 1 at 16. Then after that 12 years, you put all 5 back in the common environment and measured them both. So significant evidence of divergence that's in excess of what you expect from drift. Right? So really simple application of this for laboratory things. So that's Lande's constant variance test. Well, you can so you can look at the population cage structure. These were raised in giant open population cages, and so the argument you can turn this argument on its side. And of course, the issue with any of these tests is, well, maybe you've overestimated effective population size. If you have, then you may bias the test against drift. So one thing you can simply do is you can then simply keep on decreasing this effective population size until that number no longer becomes significant. And that gives you the converse, which is, here's the population size above which I can no longer, drift won't account for it. Below that, drift will account for it, because smaller populations, things diverge more. You get a critical population size, and above that population size, the divergence is too much relative to drift. If that population size turns out, let's say, to be 50, you know how big your cages are, you can rule out that the effective population size was ever that small. He could have, but this is back in remember, these are giant cages running for 12 years. So if you're concerned that NE is an overestimate, you can plug in different values and see how low it has to get for drift to account for these data. I haven't done it, but it's straightforward to do. It's much smaller, right? Because basically, you've got to keep on increasing this till that thing becomes 4. So eyeballing it means you've got to basically drop this in the order of 100 for you to have an effect. So the amount of divergence you'd have to have, this amount of divergence, you'd have to have an effective population size of 100 or less to be consistent with that amount of divergence being due to drift. Here, I'm testing a specific value, but you can fiddle that value and run it in reverse. So the constant variance test basically takes a population with the initial variation and assumes that subpopulations are simply partitioning those initial allele frequencies at random, that no new stuff is coming in. And that's good for a short time scale. Right here, it's basically 150 over 1,000. That's a short time scale. Mutation's probably not critically important here. What about longer time scales, where mutation can introduce new variation? So for a quantitative trait, something like a mutation rate doesn't make any sense. For a nucleotide, it does. Does that nucleotide change or not? You can get mutation rates of 10 to the minus 9 for SNPs, 1 in 1,000 for STRs. What about, though, for a trait? We have to have 2 things. Number 1, a mutation rate. Number 2, a change in allelic effect. Right? So it's not sufficient to tell you mutation rates. I've also got to tell you when you get a mutation, what's the expected change in allelic effect we see. That gives you something called the mutational variance, which I'll write as VM. You can see why I don't like to use PowerPoint for this. It'll also appear in some of the stuff I've copied from text as sigma squared sub mu. This actually turns out to be straightforward to estimate. You can do it a couple ways. Number 1 is you can take a population that's maybe gone through several bottlenecks, so it's inbred, and then make a series of lines and look at the divergence between those lines, right? The per generation divergence is the mutational variance. And these are, straightforward but laborious. When you do that, you see for a large number of traits, the ratio of mutational variance in terms of environmental variance is about 1 in 1,000. And this is pretty good. This is over a lot of traits. This value is pretty consistent. So we can then basically ask what happens when drift is removing variation and mutation is pumping in new variation. And what you can show is the between population variance at time t is this. So there's the component. The change is due entirely to new mutation. This component here, for example, let's let new mutation be 0. This is just 2 added variance times that. That's our previous result, right? That's f of t. So this here basically says, this, as we'll see, is the equilibrium variance. So if the initial and equilibrium variances are identical, you simply diverge at 2 mutational variants per generation. So we have a couple things here. At equilibrium, the added variance goes from its initial value to 2ne times the added variance, times mutational variance. The divergence per generation is simply 2 mutational variants. Notice, by the way, the connections. That's the expected added variance in a neutral trait. That's the expected polymorphism in a neutral allele. That's the expected divergence in a quantitative trait. That's the expected divergence in a sequence. So they both scale with NE, and the difference here is that mu in both of these now is replaced by the mutational variance. So this is a well-developed theory. Hill and Lynch and other people put together a well-developed theory for neutral trait evolution. So remember, the big distinction now is I'm now allowing mutation to occur in addition to drift removing variation. So I get some sort of equilibrium value. The factor of 2 comes in here because this is the mutation rate per diploid. So you're looking at both alleles in here. That absorbs the factor of 2, which accounts for the differences in this term right here. That's where that comes from. So now we're going to use those to develop models for drift in means. And there's 2 types of models that people typically use. The first one is Brownian motion. And, pardon me? But we'll put drift and mutation in here. Yeah, so we'll put that in there. So the Brownian motion model is this. So Brownian motion, if you make some simplifying assumptions, that you have very tiny changes, higher order moments don't exist, that the expected change over some small interval is 0, and the variance of that change is some amount b. Then under that model, you recover Brownian motion. There's some technical stuff, don't worry about that. That at time t, and let's think of this, the mean at time t is normal, centered about the mean at time 0 with variance b of t. Haven't told you what b is yet. And what you see here, as time increases, the variance gets larger and larger and larger and larger without bound. So you basically have a pointed distribution that gets squashed out over time and eventually becomes completely flat. That's the Brownian motion model. So what is this variance? Well, Lande showed it's pretty easy. That's simply the added variance over NE. And I can write that as the heritability times the phenotypic variance divided by the effective population size. Why am I doing this? Well, Lande applied this to a fossil record. How do you get an added variance in a fossil record? It's kind of hard. You, in theory, could get it if you saw clutches of individuals and you were relatives, in theory. But Lande said, look, it's easy in a fossil record, because remember, a fossil record isn't one dinosaur here and then, one dinosaur, 10 feet later in the strata. These are microfossils. So there are hundreds of fossils, hundreds of individuals in any single section. Show a picture of that in a second. So because of that, you can actually estimate a phenotypic variance pretty easily. And then you simply use a heritability value for a typical trait, let's say like 0.3, and see how well it fits. And the idea on these tests is you basically ask, how small does the effective population size have to be for that divergence to be accounted for by drift? If it seems unlikely the effective population size is that small from other information you have, then you can reject drift. If it's reasonable the effective population size is that smaller, then that observed pattern of change can be entirely due to drift. So that's Lande. Lande basically assumed that this variance, this is basically the change in means, is basically given by added variation over NE. Turelli, Gillespie, And Lande later came along and said, wait a minute now, we know the additive variance at mutation drift equilibrium is 2Ne times the mutational variance. So if you plug that in here, just put that in there, you now get that you can express your variance either as this expression, added variance over NE, or you can express your variance as simply twice the mutational variance. So two different versions of this test, depending upon if you have to assume heritability or you assume a mutational variance. We'll get to that. But here's the issue though. The observation is that people have looked at the mutational variance in probably about 24 traits. And over those 24 very broad traits, it's relatively constant. It's all on the order of 1 over the environment. It's all near about 1,000th of the environmental variation. So if you see a value much bigger or much less than that, that's a little bit unusual. You might argue, well, dinosaurs didn't have the things we do, but this is over a wide variety of traits and a wide variety of origins. People see this rough relationship. Again, what you're trying to do is, how can you make these tests if you can't do anything in terms of manipulating the genetics? This is kind of the best information we have. So we can first of all do a test based upon D. D is the absolute distance. So I've got two populations I'm comparing. So if I let U denote a unit normal, mean 0, variance 1, then the probability that the absolute value of U lies between about -2 and +2 is about 5%. So what I can do is I can take my observed difference between so I can do this two ways. Let's do it this way. Let's do it taking a mean at time 0 and some amount of divergence at time t later. That gives me 2 time points. I can then divide that by the expected variance of those. That gives me a absolute unit normal. And then I can basically rearrange this and say that if my absolute deviation that I see, absolute divergence, is bigger than about 2 times the square root of the expected variance here, expected variance being given by this, then I've got divergence that's larger than I expect under drift. So again, we're always coming back to these issues of, I have a pattern of divergence, I then use something to make an expected a guess at what actually better than the guess. I use that then I use other information to estimate what I expect the drift variance to be. That often requires either knowing the additive variance or the heritability or the effective population size. And then I can use that to basically ask, is an observed pattern too extreme? So here's actually the example of that. Let me give an example and I'll come back. Actually, jumping ahead too much. So tests of divergence then start with some assumed value for the effective population size or the mutational variance. Excessive divergence, so starting point, end point. Excess is divergence, indicating directional selection. If NE is small, drift can account for that. If the mutational variance is large, drift can account for that. So if I have a given value of NE or a given value for drift, directional selection is suggested if there's too much divergence. So the assumed NE is too large or the assumed mutational variance is too small. I can do the flip thing and flip that around and basically ask if there's too little divergence. That also puts bounds on the population size or the mutational variance. Next slide, I'll give an example to make it a little bit more concrete. So basically, this is just rearranging this expression up here. This expression down here, so remember this square root of the variance, that's the expected variance. So time, that's the estimate of the additive variance, so simply heritability divided by NE. So what this, you can then rearrange this and basically say, if I divide the divergence and express it in terms of phenotypic standard deviations, that's what that is. NE then has to be less than basically about 4t times the heritability in phenotypic standard deviations in order for drift to account for that amount of divergence. If your actual NE is bigger than this critical value, you can rule out that. So here's the application that Lande used for that. So these are forams. They're microscopic, hard-shelled fossils. So as you see, you can get thousands in a small fossil sample. You can basically measure a bunch of things. I can compute a phenotypic variance on this sample. And so using a data set, what he observed, what the data set observed, was about a 1.5 standard deviation change over about 500,000 generations. If you take a heritability for a typical morphological trait, which is 0.3, and plug that into the equation, plugging stuff in, there he goes, there's that constant. T is this value up here. There's the assumed heritability 0.3. There's the divergence in phenotypic standard deviations. You see that to get this amount of divergence, you have to have an effective population size of about 250,000 or less. Now, suppose you say, wait a minute now, my heritability is higher. Well, if heritability is higher, what happens is the effective population size gets bigger, its heritability gets bigger. If your heritability is lower, though, then the effective population size maybe is only 100,000, not 200,000. So the nice thing about this, even though we don't know the heritability, is heritabilities typically fall between about 0.2 and about 0.5 or 0.6. You don't typically have heritabilities near 1. A low heritability here doesn't help you because the lower your heritability, the less likely drift is. So when you do this, paleontological data suggests that the effective population size is bigger than 500,000. It's on the order of millions. Now again, you can argue that, but the point here is that that's your critical effective population size. If you assume heritability of 0.5, this goes up to 400,000, 0.7, 600,000, 1, which never occurs, 900,000. So as you see, as you tweak heritability, you still have an effective population size that if you're above 1 million in effective population size, it's too much divergence relative to drift. So of course the issue with these tests are, well, you know, we all know that effective population size depends upon bottlenecks, and what you see in terms of billion individuals in your sample, I may have an effective population size that's much less than that. Lande's tests can't get around that. What they can basically do is say, what effective population size do you have to be below for this amount of divergence that you see to be generated by drift alone? So Lande's test here is based on this constant variance assumption. So we then basically plug in either added variation, which we don't know, or heritability times phenotypic variation. That's simply the added variation. So we basically have to have an estimate of heritability, and that gives us a value of NE. Turelli's test isn't based on that. It's based upon the mutational variance. So you can go ahead and do Turelli's test, and it's built around mutational variance, not heritability. And this test, here in Lande's test, I use the absolute difference. Here, I use the actual variance. So now this is a chi-square test. I can get values where I have too much or too little divergence. Again, look at the notes for details. So what Turelli did was he went back and took the exact same data and then plugged in, and his analysis basically says that if your mutational heritability is bigger than this amount, you can account for that pattern by drift alone. Remember, as mutational variation gets bigger, you get more divergence under neutrality. So I want to get a critical level, right, below which I can't account for the pattern of variation I see, the divergence, and above which I can. And their estimate was 2 times 10 to the minus 7. The mutational heritability is simply the mutational variance expressed in environmental terms, and for most traits, that's about 10 to the minus 3. So here, I fail to reject. Why? Because commonly observed values for mutational heritability can easily generate that amount of divergence. Now, there's a problem with the test, and the problem with the test is it assumes the mutational variance becomes this, 2NE, so that's the equilibrium added variance. If you fiddle with that, if that has a value typically of this, once your effective population size is bigger than a couple of thousand, this thing gives you a heritability very close to 1. So this thing is too large in large populations. And that probably arises because mutational heritabilities are estimated in very tiny populations where there's no effective selection, very little effective selection. As my population size gets bigger, alleles that contribute to the trait are also going to have deleterious side effects, and therefore their frequency will be reduced. So this value, I think, is best regarded as an upper bound on the additive variance, and therefore this thing I don't think scales with NE as is often assumed. So because of that, I like Lande's test better, because I think NE is a more natural thing to interpret. So those are the two tests which are used when I have mutation and variation. Lande's, they basically buckets do the same thing. They basically ask, is an observed amount of divergence I see, either a between group variance or a difference between means, is that excessive relative to either my assumed value of effective population size? Smaller effective population size means larger expected neutral variance, or relative to the neutral, for the quantitative mutational rate. As that gets bigger, I expect more divergence. So I have a threshold where NE is, if it's above a certain threshold, I can't account for the pattern for drift. I get a mutational variation. If mutational variation is below that threshold, I can't account for that value for drift. So for example, in here, if this value here was 0.1, I have too little mutational variation to account for the pattern I see. So that's, we talked about Lande's constant variance test, drift alone. We've talked about these two tests, which are reparameterizations of the tame test for drift plus mutation. All these simply use phenotypic data. So questions on those so far? Another model that arises when analyzing how traits change over time is an Ornstein-Uhlenbeck model. Actually, how would you pronounce that, Wolfgang? Ornstein and then Uhlenbeck. Okay. So my non-European accent can't get it right, but OU models. And the idea here, this is Brownian motion. So this part here, so this is the expected change in variance in a given generation. That's got this constant value, that's Brownian motion. But it's Brownian motion now where I've got a rubber band attached at some point. It could be the origin. Here I'm assuming it's theta. So I've got this point theta. As the particle drifts further away from it, I've got more linear pressure restoring it back here. If I get below it, I have pressure moving it this way. So basically it's a random walk that's constrained to kind of force back to either the origin or more generally this. And this comes up in models of stabilizing selection. We imagine some optimal trait value. The optimal value is set at theta. You can then compute those models, and the chapter 12 notes I gave you tell you how to get those. And what you can show for this model is, again, the mean at time t is normally distributed, but now it's not, the mean doesn't stay expected value as the initial value. It basically goes to this value theta here. So if you plug in t equals 0, you get theta, initial value minus theta. So the initial mean is there, but as you run this, it eventually settles on theta. The second thing, though, is this variance doesn't blow up, right? Remember under Brownian motion, there's Brownian motion. The variance just kept on increasing with t. Here, under Ornstein-Uhlenbeck, the variance comes to a value which is basically the strength of drift divided by the strength of this restoring force. So here is what the variance looks like over time in Ornstein-Uhlenbeck. And what you see is that it basically levels off. So what does this say? If I've got a series of populations that are diverging, that they won't continue to diverge linearly, but at some point they'll come, and as you look at longer and longer and longer times, you won't see any more divergence. This, of course, is assuming you have the same pattern of selection on those. And to contrast, here's the variance that is, again, think of this as divergence between populations. Under Brownian motion, you have a linear increase. Under Ornstein-Uhlenbeck, indistinguishable, and then it basically flattens out. So these, Ornstein-Uhlenbeck is often used as a model for, let's say, constructing a phylogeny where you might have stabilizing selection. An application of that to expression data was Dan Hartle's lab, looked at 7 species of Drosophila. And you can do things like correct for the phylogeny. We're not going to worry about that, but you can do that. And in those 7 species, what he basically did was he fit a model with Brownian motion, those 7 means. Then he fit a model likely with Ornstein-Uhlenbeck, and he basically found that this, the restoring parameter here, so under Brownian motion, that term is 0. When he fit the model, he found that term was significantly different from 0. And he also observed that the divergence variance doesn't increase linearly with time in the clade, but rather levels off. So this fit to the Ornstein-Uhlenbeck suggests for at least these 7 species of Drosophila, for the expression levels they're looking at in genes, that those expression levels don't diverge linearly, but rather they seem to be under some sort of constraint. Because they show divergence, but that divergence sort of levels off the time. So again, that's another way. This would be the prediction under drift alone. This is a prediction under stabilizing selection with drift generating some variation about that. How much variation? It depends upon the relative strength of drift to the restoring force. No, not back mutation. This is stabilizing selection. So, by the way, you can also, in B, you can also put in mutation as well. So the way to handle that is just to assume you have the mutation drift equilibrium. So if I've got a Gaussian stabilizing selection, right, that restoring force basically is my current population mean minus the value I'm at, and then scale by the strength of selection. Chapter 12 gives you the expression for that. So that's basically, if I have stabilizing selection, if my mean gets above it, I get pushed back. If my mean gets below it, I get pushed up. So selection is generating that stability. So all these models have been models where all I do is look at phenotypes. I need other stuff. I need either a heritability or an additive variance, but I look and see how trait means diverge over time. I estimate that with an absolute difference or between group variance. I then compare that expression to the expression I expect under Brownian motion, or maybe I compare that to an Ornstein-Uhlenbeck. So that's just simply gathering phenotypes. These models can become tests for neutrality, and traits can get more information and use that. So the next thing going from the phenotypic models, and by the way, something you didn't talk about, there's a whole theory the paleontologists have developed whether or not an observed time series in a fossil record is too great or too small. And chapter 12 talks about those. There are ways you can do that sort of independent of any genetics by sort of looking at the size of the longest and the smallest walks in those. The next thing we can do is add more information. So FST, QST tests are very popular, I think improperly so. So remember that FST is a measure of variation. So if I've got a whole bunch of molecular data gathered from a series of demes, I can then ask how much of the total variation when I lump all the demes together is due to between group differences. That's FST. I can make an analogous expression called QST for quantitative genetic variation. And Tim Prout was the person that came up with this idea, but Ken Spitz, who was Mike Lynch's students, was one that named this QST. So basically what you can show is that the genetic within group variance you can write as this expression here. QST is essentially how much the additive variation is due to between group differences as opposed to total differences. There's the within group value, there's the between group value. If this bothers you, think about 2F, right? That was our divergence in here. You can then set these, you can then basically set each of these equal to total variance, rearrange and solve for this, and there's the expression you get. So it's a lot of work to measure QST. You've got to do crosses, look at within population variation, grow them in a common garden, look at between population variation. So it's much more work than simply gathering phenotypes. The interpretation here is if the QST is bigger than FST, you've got divergent selection. That is, the amount of divergence in the quantitative trait exceeds the amount of divergence in the molecular data. And the idea is you get that in the quantitative trait because you're basically having divergent selection. Some deems favor for high values, some deem for other values. The molecular data for FST are random markers not at all involved in the trait. They kind of tell you what the population structure should be. If these are equal, you can't say much. If QST is less, you can have convergent selection that is less divergence than you expect by drift. So FST varies between populations if they're neutral alleles simply by drift and mutation. QST bigger than that means you get an excess amount of divergence. So it's an idea that's widely used. And the logic behind it is additive variation scales as that, P1 minus P. FST also scales as that. One big problem is if I have dominance, additive variance is this plus some other term involving 2P over here, so it scales differently. So additive variance no longer scales as P times 1 minus P, and these two can differ even if you have neutrality. There's a big debate as to whether it differs in a consistent direction. In my mind, that hasn't been resolved. Some people argue vehemently that it doesn't matter that much, the differences are small, but nonetheless, it's something to be worried about. But there are other issues with QST. The other big thing is you've got to have genetic variances. Very often, this is done using phenotypic variances. This is called PST. So what people do for this expression is they go out and they measure a series of populations. This expression here is the between mean variance and between mean difference. This expression down here is the phenotypic variance within a group. That's absolutely dead wrong. It's dead wrong because you're confounding environmental factors with genetic factors. So if you look at between group differences, and these are often not measured in a common environment because looking at natural populations, well, that could be because you have environment 1, environment 2, environment 3, environment 4. So FST, where you don't use genetic data, and typically what happens is people look at added variation within and then get the between group by simply looking at phenotypic variances. That's wrong. And the extreme people get everything with phenotypes, and they call this PST. That doesn't work. You see it in the literature a lot. Don't do it. So these things are a lot of work to get out. So what did the data show? This is a large meta-analysis, references in that chapter 12. Here's the FST equals QST line. Look at that. So if these were equal, you expect them, 50-50, but you see a great excess of QST values. So does this suggest widespread selection? My God, there's selection everywhere. People get excited when they do this. And Mike Whitlock, who actually taught in the first course here, was one of the people who really has done a lot of analysis of QST. And the references that are given on the QST review in chapter 12 you can access. That's a really nice review of all the pitfalls. I'm only covering a small number. But he points out that there's a big problem with ascertainment bias. And the issue is, suppose I have a random variable that's centered about 0. It can be positive or negative, right? But suppose I only pick individuals which are big. It's a non-random sampling from that. It's called ascertainment bias. I pick an individual because they stand out. I don't pick them at random. And Whitlock has argued that, you can always take a set of traits which are entirely neutral and additive and simply choose a subset that has a higher than average QST value, and therefore they'll have a value of QST bigger than FST. Traits chosen this way cannot be used to make interpretations about if selection varies over those subpopulations. And furthermore, I think he correctly argues that, quote, this is a direct quote from him, makes one wonder whether traits are in fact always chosen with the knowledge that they're excessive. So if you're an ecologist, you go in and you see a bunch of excessive traits, you do QST, lo and behold, wow, they're excessive. We've already, by choosing excessive traits beforehand, ensure they're going to be excessive. So the issue here is I'm not at all a big fan of QST. I think there are problems on many different levels. You can do it correctly. And the notes talk about Whitlock's suggestions how to do this correctly. But just be aware of that. So how many of you have seen FST, QST, or comparisons, or read about them in literature? A couple of people. So now you know why I'm not a big fan. Now there are other people that are much more positive. So just be aware of that. And I think I'm pretty even-handed in chapter 12 where I kind of discussed the pros and cons for these. FST-based approaches in general for testing selection on molecular data and also these sort of tests, they're highly controversial. And they're highly controversial because people want them to work. It's easy data to get, it's ecologically relevant, and I think people's enthusiasm for wanting these to work often overrides their common sense about whether they're correct or not. So now we're in the last category of tests. Let's kind of review. We've talked about, again, the big problem is I want to see if I can detect excessive divergence. I measure divergence by either an absolute difference between the means or a between mean variance. We showed Lande's test, his constant variance test, which simply assumes I've got a population that's acting only under drift, no mutational input. I ask if an observed amount of divergent is too large relative to my assumed effective population size. We then had tests where I had longer time scales, so I had mutation and drift both operating. I then ask either in Lande's version, the mutation drift equilibrium version, I ask if the observed divergence is too large relative to our effective population size. So I estimated an effective population size that could generate the pattern with drift alone. If your effective population size is below that, drift can account for it. If it's above that, it's too excessive relative to drift. The Turelli version of that was I can get drift by having a small population, or I can get divergence by having a large mutational variance. So Turelli's is look at the mutational variance. If that mutational variance is too small, I'm not generating enough variance to account for the divergence. So both those are tests based on looking at longer timescales, and they're based upon phenotypes. We talked about FST, QST. That requires more information. The final step is, suppose I've got QTLs. I've got traits of interest, and I've got QTLs for them. And I've got estimates of QTL effects. Can I use those to make inferences about selection? So I'm not looking at the sequence pattern around the QTLs, which is what Wolfgang's stuff would talk about. I'm not looking at the site frequency spectrum or the haplotype length or the FST around QTL. I'm simply looking at its effect. So Alan Orr was the one that proposed this, and he had two versions. So here's, the idea is very simple, and a lot of tests which I lump under QTL tests have this spirit. And the spirit is, if divergence is entirely due to drift, then the distribution of effects should be random. I've got a whole bunch of QTLs, and I should equally likely pick a positive or negative QTL if drift has no impact, if selection has no impact. So Orr has two versions of this test. Number one, he assumes that the QTL effects have equal magnitude. This is his QTL sign test for equal effects. More generally, he assumes they come from some distribution. This is just a sign test. The idea is to test. I've got two lines. I make a cross. I look at QTLs. I assign the QTLs to, population 1 or population 2. I look at their effect. Obviously, one line will be big. I simply go in and ask, in that big line, I see a certain number of plus alleles and a certain number of minus alleles. If I condition on looking at the big line, which means I've got more plus than minus alleles, is the amount of plus alleles over minus alleles excessive once I condition on that? I'm going to show an example on this. I think the example is based, look at this. So here's, and I'll come back to this. So here's the application he had for it. So there's data in Drosophila. So in insects, male genitalia are scleroterized, and structures on them are really important in taxonomy. So for example, there are lots of moths in Arizona I can only tell apart by what we call pulling the tail. You take them off, take the tail off, look at the genitalia, and look for certain structures. There's a nice experiment where people crossed a couple of close lines in Drosophila and looked at genital characters. And what they saw was that of the 8 detected QTLs that affected posterior lobe, that's basically a process on that, made it larger or smaller, none of them were antagonistic. So in the line that had the larger, so I'm crossing two species, large lobe, small lobe. Obviously the large lobe is going to have more plus alleles than minus alleles. Why? Because I'm conditioning on it being large. We'll come back and show how you get those statistics. So in the large lobe, what they saw were all 8 alleles that you detected were up alleles. So given I have 5 or more up alleles in the large lobe line, what's the chance of getting the 8 of them? So all 8 are up. You can work out what it is. It's about 1%. So in this test, you have an excess of plus alleles, even in the high line. So you condition on it, remove the fact that larger lines are going to have them. So this suggests then that these are driven by selection. So here's the logic behind that. So the logic behind that is, if I have equal effects, the high line must have at least square bracket n over 2 equal plus alleles, where this thing basically is, if n is even, for example, 8, I've got to have 5 or more plus alleles. Why? It means 5 out of the 8, because I'm conditioning on the fact that it's larger, it's going to have an excess of plus alleles. You can then work out what that is, and so the probability of, by chance, of seeing this plus alleles conditioned on the fact you're in a large line. This is the equal effect. Gives you this simple expression, which is the ratio of combinatorial things. A key aside is you need at least 6 QTLs for this to work, because if you've got if you only see 6, you get about a 5% chance. If you've got 5 QTLs, you just don't get, this thing can never get less than 0.05. So the idea is, don't worry about the technical details. Those are in the notes if you're interested. I've got a large line. I got a small line. Who's got more plus alleles if the effects are equal? Obviously the large line. But what I want to ask is, given that I expect to have more plus alleles because I'm in a large line, given that fact, do I still see an excess? So what this probability here does is it says conditioned on the fact that I've got to have more plus alleles than minus alleles, what's the chance I actually see that? And as we saw in this example here, if you have all 8 plus and no minus alleles, you expect 5, 6, 7, or 8 in the high line. What's the chance of seeing all 8 as opposed to seeing 5, 6, 7, or 8 plus alleles? There it is, it's 1%. So that's an application of the sign test of equal effects. And you know, again, whether you want to use equal effect sign test is sort of unclear, but a key idea here, just like with FST, is trait ascertainment. So Anderson and Slatkin basically found that if you, again, unconsciously chose traits showing excessive divergence, then you're conditioning not only on them being the high line having plus alleles, but having a lot of plus alleles. And again, the notes talk about that. So in these tests, you've got to be very careful about how you choose the traits. You have to pick the traits at random. You can't simply look at something and say, wow, that trait's pretty big. Let me see if it fits the QTL test. Because in that case, you'll almost always get a significant result. Now, the way to get around that, and the way to account for the fact you could have unequal effects, is the QTL sign test. Not for equal effects, just the QTL sign test. And again, the idea is the same. Suppose that the difference between the two lines is some amount R. Actually, let's go back a step further. So suppose my high line has a value R. And suppose that I sample QTL effects from a distribution. We'll get to that in a second. Could be plus or minus. And I take a sample, and I keep on drawing QTL effects until I get their sum to be R or bigger, then I stop. And then in that sample, I ask, oh, how many plus alleles do I have? Then I do that again and again and again and again, and I generate a distribution. The sign test basically says, condition on this amount of excess in the plus line, is the amount of plus alleles I see excessive? How can you do this? Well, number one is I can simply draw the alleles from some presumed distribution whose mean may be given by the average effect, or I think a better way, number two, is you have a distribution of all the QTL effects, right? Empirical distribution for this study. You pick one at random, randomly assign it a sign, stick it in, pick another one, replace it, pick one random, randomly assign a sign, stick it in. That's called bootstrapping. So a bootstrap sample, sampling from replacement, is a way you can generate this where you're not making any assumptions about the distribution of the QTL effects. So you can either draw from a known distribution or do a bootstrap sample. Sounds rather esoteric, but here are two studies. So a cool study, how many know about cichlid diversity in the African rift lakes? So a really, really cool system is look at the Rift Valley of Africa, kind of East Africa. There's a series of obviously big lakes there, right? You know, Victoria, et cetera. And in those, they have cichlid fish, type of fish. Thousands of species. It's a massive, massive speciation. In the same lake, you could have a couple hundred to 1,000 different species. So people have been studying it for a while. And what they did here was they went to Lake Malawi, and they basically then took two species that differed in feeding morphology, crossed them, found QTLs. The problem is any particular trait had less than 6 QTLs. You can't apply Orr's test. Sample size is too small, so they pooled traits. So what they found was that if they looked at QTLs for jaw and teeth features, that in the high line, only 4 of the 46 QTLs were in the opposite direction. That's a highly significant value when they do this drawing values out of a distribution. So it looks like the QTL data suggests the amount of divergence in jaw and teeth traits here couldn't be accounted for by drift. Another example, and this is tomatoes for you, Wolfgang, is they looked at leaf-related traits in tomatoes thought to be involved in precipitation. So what they found was that no significant in 2 leaf and 2 trichome, leaf hair traits, but a significant departure in 2 stomatal traits, things that close the stoma. They computed p-values using both methods, is assuming equal effects and this more general, and they found the more general sign test was more conservative, giving p-values about twice as large, but still conservative. So the bottom line isn't the details, because details you have to look up. The bottom line is their approach is to use QTLs to estimate the amount to see if whether an observed pattern of QTLs you see is excessive, even when you condition on the fact that one line is going to be larger. And these basically use the idea that the effect should be randomly distributed, everything else being equal, if there's only drift involved. So those are QTL sign tests. The last thing is let's then apply these to divergence. So as I mentioned, gene expression is a very popular trait. I already told you about Ornstein-Uhlenbeck versus Brownian Motion models. Here are 2 other approaches you can use, comparing the within versus between group variance. And again, all the details and references for these are on that chapter I gave you. So here is the expected, that's the expected between group difference. That's the expected within group difference. These are the sampling variances. These are the fact you're drawing from a chi-square. So if I have basically KL populations I'm comparing, and I've got K individuals to get within lines, if I look at the ratio of these 2, what happens? That cancels out, and I simply get the ratio of those 2 variances is the effective population size divided by the divergence time, and that's an F distribution. Chi-square divided by its degrees of freedom, chi-square by its degrees of freedom. This is a variation of the mutation drift equilibrium test of Lande, and the application here was, I think this was Whiteslab at Yale that did this. It's in the notes. So basically they had 2 Drosophila species, melanogaster and simulans, so 2 comparisons. They used 4 melanogaster lines for the in-line differences. They computed the critical values here, which would be 1, 3, upper and lower values. The melanogaster effective population size was 3 million. The number of generations between melanogaster and simulans was about 50 million. The melanogaster and Yakuba was about, again, this figure here. So you can then look at mean divergence in melanogaster and simulans, and basically if that value was this or below for your data, then you could account for it by drift. Same thing for Yakuba. When they did this, they threw out a bunch of genes, and they found that of the 530 genes which are left, because they showed either consistent or non-consistent patterns, 460 were with drift, 63 were consistent with excessive divergence. So again, a within versus between comparison. But the key here is now the trait isn't bone length or wing size, it's molecular data. So one of the things that's been happening last 10 years or so is all the quantitative genetic machinery has moved over from measuring morphology into measuring genomic traits. And gene expression has kind of been leading the way. So one more thing, again, to remind you a couple of terminology, cis and trans. The classic definition of cis is I have a DNA sequence and I have a gene here whose expression I care about. Something acting in cis means that I've got to be physically on that chromosome that's affecting this allele. The idea is I've got a DNA binding factor comes in like a promoter site. A trans effect is I've got something on this other chromosome or something unlinked to it or loosely linked, that then diffuses in and basically then interacts with here. So the classic definition of cis is things like DNA binding sites. Trans are things like diffusible proteins. Well known, well understood. The problem is people then went back and started mapping what are called eQTLs. I take yeast, I take a gene of interest, gene 4. I notice a gene 4 expression level varies over individuals. So what I then do is I treat that variation as a trait. And then say for this trait that varies over individuals, where are the QTLs that control it? And those QTLs are called expression QTLs. And obviously, I've got lots and lots of transcripts. Each transcript is a different trait. That's why people like using transcript data for quantitative genetics. So I take that first trait, and then if my QTL maps close to it, that's often called a cis effect. If my QTL maps far away, it's often called the trans effect. That's approximation. The better way to call it is local, meaning if I've got a sequence of interest, if an eQTL maps around that, that is, its confidence limits can't be separated from the sequence of interest. Again, I've got a gene who's just expressed. That's the trait, and I've got sites that control that expression. You might imagine a lot of those are in the gene itself, but those could be further away. If that QTL peak covers the transcript, it's called the cis eQTL. It's better to call local QTL. If that QTL confidence interval doesn't cover it, it's called the trans, better to be called distant QTL. Very often they're on different chromosomes, it's clear cut. On the same chromosome, something in distance can still act in cis, the DNA bends over. One more bit of evidence, and we can apply this to the data, is you often see what's called allele-specific expression. So if I've got a heterozygote, oftentimes, not always, but oftentimes, you can distinguish the two RNAs, the two proteins which are made. And that's really cool because you can basically say, okay, is allele 1 over or underexpressed? Is allele 2 over or underexpressed? So a lot of studies looking for neutrality then have focused on alleles that show allele-specific expression. Why? Because you can track those alleles around. And this allows us to formally distinguish true cis and trans as opposed to this eQTL study. And how you can do that is, suppose I've got parental lines. And let's do this. Suppose I have the white allele in the white parental line is upregulated. Then I've got some other black allele in the black line that's downregulated. I make the hybrid. Okay? I can look at the regulation of the white, regulation of this. If the regulation in the hybrid is the same as the regulation in the parent, that tells me cis acting factors are important. If the regulation in the hybrid is not the same as the parent, it means I've got a transacting factor coming from the parent and diffusing it in. So by looking at allele-specific expression in hybrids as opposed to parents, we can distinguish cis and trans. We can actually quantify it because it's a little bit of both. The notes talk about that. So these are used in tests. So 2 slides. So number 1 is you can use eQTLs directly. The problem is that if you just look at eQTLs for a given transcript, there simply aren't enough. You need 6 or more. So what people do is they take families of transcripts. Here are all the genes that are involved in sporulation. Let's look at all the QTLs for the genes involved in sporulation, and let's look at line 1 and look at the eQTLs, look at their effects. Let's look at line 2, look at their effects, and then simply do a sign test by pooling those. And when this is done comparing Saccharomyces cerevisiae and a close relative, a couple things. Number 1, you need the eQTLs to be independent. If I have a master regulator, and that master regulator upregulates everything at all the sporulation genes, that's one event, not K events. So the way they got around this is they used only things that mapped in cis. Since they map in cis, they can only apply locally. When they did this, and they used allele-specific expression to get that, they noticed an excessive number of upregulated alleles or allele-specific expression, one relative to another. So they actually found evidence then of the sign test gave you a significant departure from neutrality. The last example, yes, question. Well, so the thing, yeah. So the idea is people use what are called GO, genetic gene ontologies, and they combine GO categories or KEGG categories. And again, this is kind of crude and approximate, and it's better than nothing. I think you can fault a lot of these, but it's also insightful to do that. Independent in terms of what sense? A cascade thing, and that's why that's a good point. That's why they use these cis regulators to make sure In this analysis, they only looked at those things that showed allele-specific expression and only looked at those things that then showed this factor that in a hybrid, its regulation was the same as its parent. And that means you're looking at cis effects. And that's the way they got around it, because that is a real problem. And this is the last slide of the course. Not the last video, but the last slide of the course. So you can also do, this is a McDonald-Kreitman or an HKA type test, right? So what they basically did here is they, again, looked at yeast and another relative. And what they had was they had data for, if you look at polymorphic sites, which are cis or trans, and look at divergent sites, which are cis or trans. These are within cerevisiae. These are between expression in cerevisiae and paradoxus. You can then look at these, and obviously as in a McDonald-Kreitman or HKA test, that's a significant difference. So again, you see that people working on expression data have reached down and picked a lot of these tests in to go ahead and try to look for differences or departures from non-randomness. Let me play one video to sort of close it off and then see if there are questions. And this video is in honor of Wolfgang. It's called The German Coast Guard. I've told this to one or two people. There it is. You can interpret this for us.
Das hier ist mein Sektor. Das hier ist das wichtigste Gerät des Küstenwächters. Das Gerät und das Gerät. Überlebensradar.
Thinking about in this course. So, you've been given a wide variety of topics in this course from several really good lectures and me. You've been exposed to many of the best people in the world and me in terms of covering these topics. To a group, I talked to all the instructors. This has been incredibly exciting. You guys have been great students. We're obviously thinking about maybe stealing some postdocs here or there. Potentially, we'll have to work on that. And it's been incredibly fun and exciting to teach here. So before that, let's see if there are questions about the stuff I covered, because I covered a bunch of stuff. It's 12:35, by the way, just to give you some idea. So we're doing good. We'll get out of here early. So again, the idea here was not to cover this at a level you'd understand everything, but to basically introduce the ideas to you. The notes are on there, but there's also a PDF called Chapter 12, which covers this in more gory detail. The first half of Chapter 12 is all the technical details about what happens when you have non-additive variance. Just skip right over that, and then the chapters in there on testing divergence are pretty self-contained. Okay? Great. Well, again, thank you all. On behalf of all the instructors, thank you for making this a really pleasant and exciting experience. And I assume you'll be in touch with us at some point. Okay, so thank you. Thank you.